Hello, my name is Blake Miller of BS in Biology at Augsburg College. I'm here to convince you that three scientists, Osamu Shimomura, Roger Tsien, and Martin Shelfie, deserve more recognition as some of the most important scientists in cell biology discoveries. I'm also here to inform you about GFP, or green fluorescent proteins, what they are, what they do, and how they are helping us every day. First, what a GFP is. They are small proteins that fluoresce whenever exposed to light in the blue to UV range. They were first isolated from the jellyfish Avictoria by Osamu Shimomura. He harvested these by essentially killing the jellyfish, tens of thousands of them, just to actually discover what GFP was, which may seem immoral to some of you, but as I continue this speech, I hope you realize that our expansion of knowledge has been at equilibrium with this loss of life. The GFP is also explained as having a major excitation peak at below the UV, or the visible spectrum, at about 400 nanometers to be exact, but can be seen, which I'll explain why in a bit. The GFP gene can also be introduced into organisms and have since been modified and are infinitely useful in understanding protein targeting and gene expression, as well as many other factors. The GFP structure, it is composed of 238 amino acids, weighs approximately 27 kilodaltons, and only about 4 of these amino acids, 4 out of the 238, directly produce the fluorescence effect. This is called a chromophore. The barrel structure keeps the chromophore away from solvents. Why it glows? Well, the Avictoria has another molecule within it, the aquarin, which fluoresces, or which bioluminesces blue light, which is then absorbed by GFP at its second excitation peak, and which lets off a green hue, which we can see. The reason we can see GFP in other organisms is because of one man's idea, Martin Shelfie, to use GFP as a marker that could be attached to a promoter. He had hoped that the GFP attached to the promoter, the GFP gene would actually be produced whenever the promoter was attached and active. Whenever the promoter attached was activated. He then incorporated the GFP into the E. coli promoter, and they fluoresce green whenever exposed to blue light. This was a huge success and opened the door to viewing gene expression in a whole new way. It was very exciting in the scientific community. Such as mice expressing GFP genes in their genome. GFP is useful is especially useful compared to other fluorescent proteins because folding occurs automatically without the assistance of enzymes. It also only requires atmospheric oxygen, so it's very green per se, and doesn't require other steps really, which is extremely important in our research. Organisms can also be genetically engineered to have these genes produced. The gene will still work without the introduction of other genes. There are, however, many problems with the wild-type GFP found in Avictoria. For example, it has broad excitation peaks, which means that it shows in multiple colors at specific wavelengths. making it unsuitable for FRET, which we will talk about later in this demonstration. It is also slow in the formation of the chromophore. It takes about 2 hours for it to fully oxidize. It also has a tendency to form dimers and trimers, increasing its molecular weight. This can inhibit the function of both the GFP and the attached protein. And it also inhibits the movement of the tagged protein through the membrane and across the cell. It also is non-optimal. It is a non-optimal codon usage for mammals and has poor folding efficiency at above room temperature. Roger Tsien created a solution for this. He engineered colonies of bacteria to express GFP and then just let them grow. This naturally produced mutant varieties of GFP, and although many of these mutants destroyed the fluorescence effect, some of them helped to improve on the problems of wild-type GFP, such as shifted emission peaks, changes in color, increased brightness, or oxidize much faster than 2 hours. These are examples of what he accomplished. The GFP drives are obvious, but the MRFP1 is a previously altered protein. The EXC here, it represents the wavelength at which the excitation occurs, and the EM represents the peak emission wavelength. If it has an E before its name, right here, it shows that this is an enhanced brightness mutation. This has helped us widely. This has infinitely helped us in many scientific fields, such as it is widely used in genetic studies to indicate the expression of other genes. And so, in both pro and eukaryotes. And so, GFP is expressed in the inhabitant, but only where the original was expressed. It can also be used to tag proteins and is used in FRET, or fluorescent resonance energy transfer, which can be used to view real-time events within the cell by using 2 different GFPs that emit light at different wavelengths when the proteins interact, such as an enzyme acting on its substrate. In conclusion, these scientists deserve more recognition for what they have accomplished. These accomplishments have been and still are being used to make new discoveries. The discovery and development of GFPs have been essential to discovery and understanding of what we know about gene expression, as well as intracellular systems, protein folding, and protein transport. They have also been helpful in understanding brain circuitry, membrane potential, and have been helpful in gene therapy. This recognition can possibly provide more funding to continue research in GFP use, which will expand our knowledge in every scientific field, which is why I believe that you should publish a story on these 3 scientists. Any questions?